Hi everyone. So we will wait for some 5 minutes. So if somebody wants to join, then they will join. After that, we will start the class.
मन चलता ही तब तक है जब तक कोशिश दो तरफा दोनों तरफ से प्रयास किया जा रहा है और अगर कोशिश जारी रहेगी तो कोई भी आपके रिश्ते को तोड़ नहीं सकता और एट दी एंड अगर हो भी जाता है भगवान ना करे उस नाते इस कदर खराब हो चुका है कि उसका ढूंढना ही एकमात्र रास्ता है उसके बाद आपको बस एक चीज याद रखनी है अपने आप में सुकून ढूंढने के अलावा कहीं भी ढूंढोगे तो एक धोखा मिलेगा और फिर आप उस धोखे का जो इल्जाम है वो सामने वाले पर डालो जबकि गलती हमारी थी हमने ये एक्सपेक्टेशन रखी हमें ऐसा मिले इस चीज से अगर आप बचेंगे तो कुछ रिश्ते आपको सबसे बड़ा रीजन ऐसा कि आपको सब संभाल लोगे क्योंकि आपको पता है मेरी खुशी और मेरे दुख का कारण सिर्फ और सिर्फ आज के वक्त में ब्रेकअप्स का सबसे बड़ा रीजन क्या है सबसे बड़ा रीजन है लोग एडजस्ट नहीं वजह तो कभी कभी कायरों को भी प्राप्त हो जाती है
So okay, huh. So we will start the class. So yeah. So first of all, this is an extra session. So which is like in which we will discuss some of your assignment questions. If you are having any doubt, first of all, you can ask if you are having any doubt. So this is like some extra session from your regular TA class. So if you are having any doubt, then you can ask. If you are not having any doubt, I have prepared some of the questions for you so that you can read the questions and you can answer that questions. Since this question I have made on the point of your exams, so I would like to, because enough questions we have discussed, so I would like if you will answer these questions. So first of all, if you are having any doubt, then you please tell me.
So what is the role of SODs, ma'am, in biological system?
SOD is in biological system. Okay, first of all,
Uh,
SOD is superoxide.
what's the role?
Okay, so first of all, SOD, as you know that,
Superoxide dimutase,
superoxide dismutase, not dimutase. This is dismutase.
dismutase.
So superoxide
Okay.
dismutase, they are kind of antioxidants. Antioxidant means,
Oh,
first of all, mitochondria. In mitochondria, lot of reactive oxygen species will be generated. Reactive oxygen
oh,
species will be like superoxide, peroxide, hydrogen peroxide. Many will be there, and these reactive oxygen species are the signaling molecules for our human body. But at some level, if you will see, some of the cells got mutated. In that case, what happen is, when the concentration of superoxide will increase in the human body, then superoxide dismutase will come into the function, and they will detoxify the excess of superoxides. So they will convert superoxide into the hydrogen peroxide by taking two electron, and again that hydrogen
okay.
peroxide will be converted into water by the catalase. So first of all,
Okay.
superoxide will be converted to H2O2 by using SODs, and then H2O2 will be converted to water by using catalysis. So basically both
Oh,
SODs and catalysis are the antioxidant enzymes.
oh. So H2O2 will finally converted into water.
Yeah. So if they will ask you, so question can be asked in two ways. So superoxides will be converted into hydrogen peroxide by taking how much electrons? Two electrons. And superoxide will be converted into water by taking how much electrons? Four electrons. So
Once
first,
again repeat it, ma'am. Please, I am noting down.
yeah, so superoxide will be converted into hydrogen peroxide, that is H2O2, by taking two electrons.
That
So
is two
super,
electrons.
so yeah, so if you
Okay,
will
ma'am.
see the reaction, if I will share one, okay, I will write
Okay,
1 minute, I will share one slide with you
okay, ma'am.
so that I can also write and you can see.
Taking notes for exam.
So slides are visible, right?
Yeah, it will be spent.
So okay, so if superoxide will be there, so superoxide what it will do is it will take two electron, two H positive
Okay.
plus two electron, and it will be converted into hydrogen peroxide, H2O2. So this reaction is catalyzed by SOD, superoxide dismutases. And
Okay.
if now H2O2 is also toxic to the human body, so H2O2 will take two more electrons, two H positive plus two electron,
Mm.
so it will be converted into water. Water, how many molecules? Two molecules of
Two molecules of water.
water. And this reaction is catalyzed by the enzyme catalysis.
Catalysis, catalysis,
Catalase.
catalysis. Okay.
Yeah. So if you will balance the reaction, what you will see, H2O2 will be cancelled out,
Mm.
superoxide plus 4 H positive
Mm. Plus four electrons.
plus 4 electron
Mm.
will be converted into two molecules of,
So totally four electrons.
yeah, so total 4 electron to convert it into the water. But if they are asking for hydrogen peroxide, then how many electron?
Four hydrogen peroxide means eight electrons.
No. You listen properly. What I am asking is, so three questions can be asked. So
Oh,
what is the role of SODs? So the role of
okay.
SOD is converting superoxide into the hydrogen peroxide. So the role of SOD is,
One minute. Role of SOD is converting the super peroxide into.
so the role of so the role of superoxide dismutase is converting superoxide into hydrogen
Mm.
peroxide.
Okay.
So this is the one statement from which question can be asked. So
Okay.
the second question is, what is the role of catalase? So the role of catalase is converting hydrogen peroxide to the water molecule. So this is the second statement from where question can come. So the role of
So
catalase,
role of catalysis is a conversion of
hydrogen
hydrogen peroxide
peroxide
into
to
water.
water.
Okay.
Okay. So the
Okay.
third questions can be, how many electrons are required to convert superoxide into hydrogen peroxide?
Two electrons.
Two electrons. So this will be the third question. So fourth question is, how many electrons are required to convert hydrogen peroxide to water? hydrogen peroxide to water. 2 electron, right?
Yes, ma'am.
So the
2 plus
four
2, totally 4.
So the question is how many electrons are required to convert superoxide into H2O2?
Superoxide into H2O2, 2 electrons.
Okay, so the next question is how many electrons are required to convert superoxide
I
into
think
water?
4 electrons
Yeah,
totally.
yeah, yes. So 4 electrons. So these four questions can be asked.
Okay.
Okay, so
Hmm.
yeah, yes. So the next doubt.
Hmm.
So if you have some doubt is there, you ask. No. So shall we discuss the questions?
Yes, ma'am. We can discuss finally and ask if you have any doubt.
Okay, no problem. So the questions are very simple. So if you know, please answer me, okay? So we will discuss if questions, if any answer you know, we will discuss the question, answer both.
Okay, ma'am.
Yeah. So we will move to the first question. So how many inorganic sulfur atoms attached to the iron center of cytochrome P450?
No idea. Sorry.
No idea. So, okay. So this you have to remember anyway. So in cytochrome P450, that first you have to remember cytochrome P450 is in
Iron,
active,
and
yeah, in active site of cytochrome P450, which metal ion is present?
iron is
Iron.
present.
Yeah, this you have
Hmm.
to remember. Anyway, in this,
Oxidation of ethylene,
yeah,
alkene
so,
to epoxide. Hmm.
yeah. So the thing is, uh, means in this question they have given, but you have to remember, like what is metals. Metal is present in cytochrome P450. First of all, it is iron. And what is the role of cytochrome P450?
That is the oxidation,
Oxidation.
epoxy oxidation,
And hydrocarbon to alcohols, right? If CH
hydrocarbons to
alcohols.
alcohols.
Yeah, so if CH will be there, if CH is there, then it will be hydrocarbon. No any heteroatom is there. So in that case, it is difficult to metabolize hydrocarbons by human body. So what
Okay,
cycle?
what is the product of alkene, ma'am? Oxidation of alkene to
Epoxide.
alkene to epoxide.
Yeah,
Okay.
alkene to epoxide and
Hmm.
hydrocarbon to alcohol. Because it is difficult to metabolize by human body, what it will do is it will give oxygen to the, like if you
Okay.
will see, if it is like C H, what it will do is it will convert C
CH3
mm,
OH. One oxygen will be added,
will
isn't
be
it?
yeah.
OH.
So what happen is the metabolism of this product will be easy by the human body.
Okay.
That's why this will be catalyzed by cytochromes. So this cytochrome is iron-based enzyme, and the function of cytochrome P450 is converting hydrocarbons to alcohols, alkene to epoxides. So the basically we will call it as monooxygenase, because
Okay.
monooxygenase means single oxygen. They will give one oxygen to the substrates.
So this is monooxygenase.
Yeah, we will call
Okay.
it as monooxygenases.
Okay.
Monooxygenase.
Okay.
Because it will give one oxygen to the substrate. So these are monooxygenase, and the
Okay.
number of inorganic sulfur atom present in this is one.
How many sulfur atom, ma'am?
One.
None.
One.
Okay.
You have to remember this.
No sulfur.
Fine.
Okay.
Yeah. So this will oxidize the substrate by converting it into by transferring it to a single oxygen atom.
Okay.
That's why we will call it monooxygenasis.
Okay.
Okay. So this is the second question. How many histidine residue present in the methane monooxygenase?
How many?
Histidine residue present in the methane monooxygenase. So first of all, in methane monooxygenase, which metal is present?
Then
Hmm?
remember sulfur. I think not sulfur protein. Monooxygenase means cytochrome, isn't it?
You take
Cytochrome.
cytochrome.
Hmm.
No, no.
Monooxygenase means cytochrome.
I am telling which metal is present in methane monooxygenase.
I think iron.
Iron. Yeah, iron. Iron is one. How many irons are present?
And what's ma'am?
How many irons are present?
Two iron.
Yeah, two irons are present and they are present in which form? Fe, oxygen, Fe. So two irons are bridged by oxygen atom. Fe, O,
Fe.
Fe. So two iron
Like
atoms,
that of hemimerthrine.
like yeah, like
Yeah.
that only. Like you see. Okay, so histidine is which amino acid? This is histidine, right?
In one,
So
two,
how many histidines are present?
two. One is 147, another one is 246.
Yes, so two histidines are present in the methane monooxygenase. You note down the active site of methane monooxygenase. Okay, I am waiting for some 2 minutes. You note down the active site because many questions will be asked on the basis of this.
What is E144 means? And what is E144?
Uh-huh.
That glutamine is on what is E amino acid? E
This
means
this is glutamic acid.
E also glutamine. This is glutamine. E means glutamine.
Glutamic acid.
Okay. Okay, ma'am, we can move to next question.
Okay, fine. So moving to the next question. So other than histidine, which amino acid is present in methane monoxygenase?
Glutamine, now only
Yes,
glutamic acid.
yes,
Now only.
yes, yes. Fine, good. Glutamic acid. So ferredoxin is two iron cluster. True or false? So in ferredoxin, how
Action is,
many irons are present?
I think, 4, Fe4S4.
Fe4S4 is cubane.
Hmm.
Fe2S2 is ferredoxin, right?
Fe2S2.
So Fe2S2 is two iron cluster. True, na?
Oh, the 4 sulfur means another 2 comes from organic.
No, no, no.
Hmm. That
No.
S histidine is there.
Uh-huh.
I can't understand. S histidine means organic sulfur.
S16 means organic sulfur.
Organic sulfur.
Huh.
So
And
we have
these
to
two,
consider only inorganic sulfur. So FeS2.
so these are inorganic.
Oh, okay.
These are organic.
Okay.
These are organic.
Okay, ma'am. Move to next question.
Fine. So this is true statement or false? Ferredoxin is two iron cluster.
True, true.
Yeah, two irons are present. Okay. So here I have explained. So ferredoxin is two iron cluster and four iron cluster. We will call it as ferredoxin. And this we having one iron atom is called rubridoxin. And this is aconitase. So this is
And Peridoxin 4 ion cluster is also there, 2 ion cluster is also there.
yes, yes, yes.
How can we differentiate that?
No, they will not ask you question like this.
Mm.
But you have to
Yeah, I thought Fe4, S4, that's why I told you Fe4,
yeah,
S4.
yeah, yeah. Now I
Mm.
remember. So these all three Fe4S4, Fe3S4, Fe2S4. These all are for ferredoxins. Whenever more than one iron atom is present, all are ferredoxins.
Oh,
Like that you can
okay,
remember.
okay.
And the cluster which is having one iron atom, they we will call it as rubridoxin.
Rubridoxin.
Fine,
Oh,
na? Now you can remember.
okay. Aconite is also one type of Peridoxin.
Ferredoxin only. But they
Okay.
have given some specific name to aconitase.
Mm.
Yeah. So which ligand attached to the close to the porphyrin ring in cytochrome P450?
Chiliganaya.
Cytochrome P450, what is active site to cytochrome P450?
Histidine, I think so. Histidine.
Okay. Cytochrome P450, which metal is present?
Iron is present.
Iron is present. So,
Mm.
pakka histidine. Histidine is present in that this na methane monooxygenase. In cytochrome P450 also histidine is present close. Pakka.
Mm. Please give the structure of cytochrome P450.
1 minute.
And
What you were telling?
structure of cytochrome P4.
So now,
Cysteine. Mm.
okay. Okay, so they will ask you the question in paper, then you will tell, show me the structure.
Yeah. Yes. And as for all the structure, then because I am not a biology student, I am purely chemistry.
Okay.
So it's very difficult to remember all these things.
Chalo.
Now
But
I am
but you
consolidating all the reports
but you were
and
telling.
table format. Table format I am preparing. That's it will be useful for me.
Yeah, yeah. And
Okay.
you see, this you have to remember cytochrome
System.
P450. Which iron
System.
you see. Now I will ask you many questions from this single slide only. So
Okay.
first of all, what is cytochrome P450? So what is metal?
Monox.
1 minute. For what metal atom is present in cytochrome
Iron.
P450? Iron. And what is the oxidation state of iron?
Fe3 plus.
Fe3 plus. Okay.
Mm.
So what is the role of cytochrome P450?
Oxidation. Single oxygen will be added with this monooxygenase.
So
The
this is not
substrate.
oxidation. This is oxygenase.
Reaction is oxidation. So it is known as monooxygenase.
Yes, so monooxygenase. So why this is happening in the human body?
Because
Because
mm,
metabolism of
more
this
water
is
cell, yeah.
difficult. Metabolism of this is difficult
Oh,
because you see human is 70% of water, right? So
oh,
there is no heteroatom in this, so metabolism of this
oh,
species is difficult. So what these enzymes will do, they will convert it into alcohols by giving
oh,
one
okay.
electrons. So the
Oh,
metabolism of it will become easy. That's why
easy,
cytochrome
easy. Okay.
P450 is helping in the metabolism. So we will
Okay.
call it as oxidative metabolism. It is a
Oh,
primary method of drug metabolism.
oh,
The purpose of drug metabolism is to make drugs more water soluble so they can be easily excreted from the human body.
okay.
Okay, out of alcohol and this which is soluble more.
Okay, okay, okay. Oxidation, that oxidizing product is soluble in water.
Yeah, yeah, that's why.
That is why that oxidative metabolism is done
Yeah,
by
yeah.
cytochrome P450. Okay, ma'am. Next question.
Hmm. Yeah, you remember this full slide is very important for even
Yes, yes, it's
MC.
very, very. Yeah, I'm only expecting this class.
Yeah.
Nobody has responded.
What?
2 times I missed that class on Sunday, but it was not happened, I think so.
Hmm, hmm, hmm.
Sunday, last Tuesday also.
Hmm, 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 hmm. Yeah. So from this paper, it is okay. From the next competitive paper, also, these classes are very important. This
Okay.
coursework is important.
Yes, yes, yes.
So the next question is metal ion
Iron,
present.
iron, iron,
Oh,
iron.
okay.
It's easy question.
Okay, okay. So active site of this also I told. Active site of particulate form of methane monoxygenase.
FeO, Fe, I think.
Yes. Now only I explained, right? This
Yeah.
this
1 minute, 1 minute.
this one,
What question? FeO, Fe, please.
right?
Yeah, here only. Yeah. So,
Yes. So binding metal for ATP is,
so binding, binding metal already discussed, I think. Mg2 plus.
yes. Binding metal for ATP is magnesium plus 2. So
Already that question.
it is an essential
1 minute,
mineral.
ma'am.
Yeah, yeah.
Yes. Okay.
Fine. Shall I move?
Yeah, next. Okay.
You remember this ATP? This ATP is
Adenosine
adenosine
triphosphate.
triphosphate. It is the main source of energy in the cells. It must bind to magnesium ion in order to be biologically active, and it is also called as magnesium ATP. Okay.
Okay.
So this question, I think you only replied me, right?
Yes, true. I think so. It's already.
It's
This
already,
question was asked.
yeah, you only explained me that it is true only.
Because that four histidine groups are there.
Ha.
That is a very bulky group.
Okay.
So that acts as a shielding group.
Hmm.
So what happens? That oxygen will be carried out in a particular that ion point only.
Okay, okay.
So it won't allow the other things to do.
Ha. Okay,
It
okay.
was explained by one faculty.
And okay. So this is true. Hana?
Yeah, yeah, 4 5 histidine groups are there. That bulky groups that act as a that protective one, shielding pocket. So they won't allow that unnecessary reactions. Only
Okay.
the ion will be directed towards oxygen.
Okay. Fine. Good. Okay. So this statement is true, Hana?
Yes, ma'am.
True. This is true. This is true. So which of the following is not a significant biological oxidizing agent? This also I think you only replied.
Actually, NAD plus is correct answer, but
Hmm.
the answer was given as NADH in that assignment questions. I got wrong My answer is wrong, they told.
So NAD positive is right,
NAD
na?
plus is the correct answer, but in the assignment NADH is given.
Hmm.
Actually, if hydrogen is there, there is a reducing agent.
Yeah,
It won't be a,
that's what. Yeah, yeah, yeah.
yeah, that assignment questions have to be rectified for all.
Mm-hmm. So yeah, so that is wrong question. I think NADH, na?
Yeah, you can. Nobody has represented, I think so.
Mm-hmm. Okay, so NAD positive is the strong oxidate, like significant.
Oh, which is, sorry, not a,
What?
not a significant biological oxidation agent. All I don't know, ma'am, answer.
They're not asking significant, na?
Oh, yeah,
Then it should be iron.
Fe32 plus.
Yeah, yeah, yeah. Oh, like that mistakes will happen.
Yeah,
I will make it correct.
it's answer. We have to put.
So the answer should be what answer should be?
I don't know that answer. I'm still, I have
Iron plus 3.
Fe32 plus.
Because oxygen is present in human body and they will oxidize. FAD and NAD both are cofactor. Okay, so they are both oxidizing agents. So Fe plus 3 is not a significant biological oxidizing agent.
But the same question was given. Instead of oxygen, NADH is given. What is a significant biological oxygen oxidizing
So
agent? The
that was
answer is
significant or
NADH.
not significant?
Okay, significant oxidizing biological agent, that is NADH is given instead of O2.
Hmm.
That is the correct answer. The other things are not significant biological oxidizing agent.
Okay.
Fe3 plus NAD plus FAD. So
Mhm.
I have doubt in this question.
Now it is clear, right?
Yeah, the question is, I think that I have doubt in this question now.
Still you have doubt.
Because, yeah, because the same question was asked like this, ma'am. Which of the following is a significant biological oxidizing agent?
So options are?
Instead of oxygen,
Hmm.
NADH is given.
Okay, okay.
Please you check it with the answer, professor.
Okay, okay.
Yeah, the question is wrong.
Okay, fine.
Okay,
But
next.
not significant, then it will be iron.
And the assignment
That,
question already asked in the sixth, fifth, I think so. Fifth assignment, fifth, the same question was asked.
okay, okay.
But with this, which of the following is a significant biological oxidizing agent? Answer is NADH. Instead of O2, it was given. The other things are not significant biological oxidizing agent. They don't.
Okay.
Please check that answer.
Yeah, yeah, I will check.
Okay, next question.
Hmm. So this question I will not tell. This is very simple. Then this is
Oxygen is no false. This triplet ground state
yes.
and border order is true.
Yeah. So this statement is true. This one
Second one is true. First one
and
is wrong.
first one is false. So
Hmm.
overall statement will be
Wrong.
wrong. Yeah, right. So this is triplet. So this is triplet oxidation, triplet ground state, bond order 2. So storage of iron in body is carried out by.
Iron and ferritin. Ferritin,
Okay.
it's given.
So transport of iron.
Ferritin is answer. Yeah. Transport of oxygen, hemoglobin, ma'am.
Okay. So
Myoglobin storage,
yeah. Storage of what?
oxygen.
Okay. So transport of iron.
Ferritin, storage of transfer of iron, I think so. Uh, cytochrome 450.
Ha ha ha. No.
Transfer
Now
of iron, wait, ma'am.
no. Now only we read. But
I wrote
cytochrome
down.
P450 is transferring iron. They are transferring oxygen.
No, no, no. That is oxidation.
That is oxidation. Not even they are not
There I have no
transferring
time.
even oxygen.
Yes, yes. Transfer of
So
iron,
okay.
which is done by.
Okay, wait. Four questions will be asked from here. Storage of iron, ferritin. Transport of iron,
Ferritin.
transferrin.
Oh, transfer of ion, transferring. Okay.
Transferrin. Storage of oxygen, hemoglobin,
Oh,
sorry, I am also. Storage of iron,
oh,
ferritin.
okay.
Transport of iron, transferrin. Then storage of oxygen, myoglobin. Transport of oxygen, hemoglobin.
Okay.
Okay, and oxidation of iron in transferrin will be plus 3.
Okay.
So these five questions you have to remember.
Iron 3,
It is,
oxidation state is iron 3, Fe3 plus.
in transferrin only.
Deoxy,
No, no.
transferring Fe3 plus.
Yeah, yeah. So transferrin will
Oh,
transport the iron in plus 3 form.
oh, okay, ma'am. Thank you.
Hmm. So transferrin is the used for transporting iron, but it will transport the iron in plus
Okay.
3 form.
Only in 3 plus form. Okay.
So the next question is this one.
This is last one is already asked, ma'am.
This is
Odd man out. That is last the heme unit with one axial histidine residue cited here.
fine, then
This
I
is
should
wrong.
not explain. It is clear.
That answer is given like that. I chosen first one that is wrong. In for assignment, I got a
Huh.
wrong answer.
Then it is
So
wrong.
last one is the correct one.
So no need to explain. You know this, right?
No, no, no. Only answer only I know.
Okay. Okay. So cytochrome C oxidase contain all but one of the following is odd one out. So first is a copper to center bridge by two cysteine residues. So if you will see this is copper, this is copper. So
Copper center.
two copper, two copper first of all. So here also two copper. This is true. And they
Okay,
are
copper,
bridged
copper distance. Okay.
bridged
Okay,
by
ma'am.
two cysteine residue. Yeah, first cysteine, second cysteine. So right only na copper
Okay.
2 bridged by two cysteine residue. Fine. A
Yes,
heme
correct. Okay, first one.
a heme unit with
Hmm.
axial histidine is, yeah, this is fine.
Axial is here. Okay.
Histidine is there.
Okay.
Then a copper center
Yes, ma'am.
coordinated by three cysteine residues. This is also fine.
One copper center is coordinated by 316. Where it is 1, 2.
Here
This is
they,
listed in where it is, ma'am.
no, no. Here it is shown just bridged. So if you will see
Here,
one,
okay, copper center
if you once
coordinated by 316.
one minute, let me tell you. So
Uh,
one copper is again coordinated to two more cysteine. Here it is not shown.
yes, ma'am.
Okay.
316 is there. What are the 316? 16 216, 16 220. Another 16 is meta. Ma'am, 316, I can't understand where is the
One minute,
316.
please. 1 minute. 1 minute. This you are telling me wrong answer. Right answer is C. This is the right answer. Odd one out I have to select. So this one is wrong.
So
So option C is correct option.
C is correct, ma'am.
Yeah.
This is the odd man out,
Yeah,
okay.
this is the odd one out because in cytochrome C oxidase,
But here only two histidines, three histidines are there. There is no three histidine.
where you are seeing, I don't
Both the
know.
answers are wrong. But in the assignment questions, the fourth one is given.
You don't tell me about the assignment. You read.
Only two histidines are there. Two cysteine, two histidines there. Both of them are wrong.
What you are talking about? They
Fourth
are
one is
they
also
are
wrong,
asking
ma'am.
about just axial. They are not asking about total histidine. First of all,
Here it's sited near a copper center coordinated by Three histidine residues given.
That's
That is
what I am trying to explain. This one you are seeing, that is not a full structure. They are here. I am just
oh,
trying to
okay, okay, okay.
explain the first point, that a copper 2 center
Oh,
bridged by two cysteine residues. A heme is present with axial histidine, but a copper center coordinated by three cysteine residues are not here. But here
oh,
there are a copper center coordinated by this. You have to this you see. Copper center is coordinated by three histidine residues, not by three cysteine residues. So three
but
histidine.
in the assignment it was given.
Yeah, I
That
don't
answer
know
was
now
given.
which assignment you are talking about. I don't know. But
That fifth assignment, ma'am. That I got the three questions wrong.
okay, but you don't means you don't mix these things. You just focus here
Okay,
only, whatever I am talking about. Then you cross check
okay, okay,
again with the assignment.
okay,
Okay. So
okay.
here cysteines they have asked. If they could have asked histidine, then it was not odd one out because histidines are
Yeah,
present.
only two.
They are
Only two cystine is there. Okay,
on they are talking about cysteines, so this is odd one out.
okay. Next question. That's all, ma'am. Question.
Next question is this one.
The pathway nucleophilic Paraxocarpus species. Nucleophilic.
Nucleophilic, right?
This assignment question. This was given.
So this is very,
Back, ma'am. Okay.
this is very easy.
This is also there. NADH. NADH2O. NADH. Yes, NADH.
Yeah,
Yeah, NADH.
this is very easy. Shall I explain or not?
Not. Do you want to explain?
No, no, it is not like that.
Right, right.
Okay,
That
this is clear, right?
yes.
Hmm.
This is also given that protoporphyrin 11 heme uh,
9.
9 multiprotein monomeric proteins. The one and the last one protoporphyrin 9. Last two is it ma'am?
Yeah, yeah, yeah.
One other confusion is there.
So the next is the metal that is non-prevalent in the biological system.
Palladium I think so.
Yes, yes, palladium. Because this is this is heavy atom.
Oh, heavy metal. Anemia na iron da.
Hmm?
Which metal deficiency anemia iron. Third one
Third.
iron.
So
Other in cancer cisplatin means
which of the call is used for treatment of
cis
ovarian
cisplatin.
carboplatin?
Hmm. Carboplatin.
No, because they are specifically asking about ovarian cancer.
Hmm. But cisplatin, what is the use, ma'am?
Cisplatin is also used for cancer only, but nowadays cisplatin is like Now, after some time, research will improve, na? So people have made oxaliplatin, carboplatin, which will target the specific kind of cancer.
In assignment, cisplatin is given now.
No, you are confusing me also.
But please, in that assignment zero, this question was asked that is in cancer. Once again, I'll check, ma'am.
You check because they are
You
asking
so
about,
many wrong answers are there.
you
Okay, ma'am.
see, ovarian cancers they are talking about,
Okay,
right?
carboplatin. Okay, ma'am.
Hmm.
Okay, carboplatin. I'll check it, ma'am. I confused. Sorry.
No, no,
Okay,
no
I'll check it.
problem.
Hmm. Okay, ma'am. Next question.
Hmm. Which of the following element causes the Alzheimer's disease?
Lead,
Lead.
lead.
Lead and cadmium.
Cadmium.
Cadmium is not in the option. Lead is there, so
Okay,
lead.
you have to note it down. Okay.
So photosystem 2 contains the metal.
Metal ion. Oh, recently we have studied here. I think manganese.
Yes.
Because chlorophyll contains manganese.
Note down these things also. Just don't remember the answers only. Okay. These things also, whatever key points I have.
Yeah. Calcium is also there.
Mhm.
Okay, next question, ma'am.
Hmm. Storage and transfer of iron in the
Actually, ferritin, but transferrin is
here
not
should
given.
not be there. Otherwise,
Yeah.
in option
The same
transferring should be
question is
there.
asked in that year also. Hemocyanin, copper, ma'am.
Huh.
Ma'am, I don't doubt what is the role of iron in the enzyme cytochrome oxidase.
Huh?
Role of iron is role of iron
Hmm.
in the enzyme cytochrome oxidase. Electron transfer,
मेरे को समझ नहीं आया मीन्स कैन यू एक्सप्लेन अगेन
role of iron
हाँ
in the cytochrome oxidase, enzyme cytochrome oxidase.
हाँ
What is
हाँ
the role of iron? Electron transfer, ma'am.
रोल ऑफ आयरन it is involved in this cytochrome C oxidase na that is I think
Uh,
iron only that
iron, that is
transferring
electron transfer is the role. What's the role of iron?
and I don't remember that exactly
Okay,
what
ma'am.
are the options.
Oxygen transport, electron transfer, ATP synthesis, DNA synthesis.
Now we will check this question.
Okay, okay.
So this active site you want to note down or you have already hemocyanin.
Methionine copper.
No, this active site. This is deoxyhemocyanin. This is oxyhemocyanin. You want to note down or it is already there? If you want to note down, then you can note.
Okay, noted. Question will be easy or tough, ma'am?
Easy, easy.
Easy. Okay. But because it's new to me, so I am afraid of. So
Mhm.
I am learning. Okay, ma'am. Next question we can
One
move.
minute, 1 minute. Can you please on hold? 1 minute.
Okay.
हेलो। हेलो, हाँ हाँ भैया बोलो। हाँ भैया बोलो। हाँ भैया। हाँ। हाँ भैया। माइक कब हो रही है भैया? हाँ भैया, आप रूम नंबर लिख दीजिए। हाँ।
Okay, ma'am. I'll check it in the. Okay.
भैया बोलिए। ओटीपी है 9274। 9274। 9274। हाँ हेलो।
Ma'am, are you here? Ma'am, ma'am. ஏங்க ஏங்க கேட்டாங்கன்னா ஆன்சர் பண்ணுங்க. ஓகே, இது என்ன பண்றது? ஏங்க, இது எப்படிங்க வெளியில வந்துச்சு? ஐயோ, ஓகே, வெளியில வந்துருச்சு. ஏங்க, இது என்னது? ஏங்க, இது தனியா வந்துருச்சு, இது தனியா வந்துருச்சு. ஏங்க, கால்லயே விழுகாத மாதிரி இருப்பீங்க, எனக்கு பயமா இருக்கு. இங்க பாருங்க, எந்த வந்துருச்சு. ஒண்ணுக்கு வந்துருச்சு.
Hello?
எப்படி மாற்றுறது? இதுங்க, இந்த இருக்கு, அதுல மாட்டணும். இது மாத்த மாட்டேங்குது. அதைய கொடுங்க. கொடுங்க, போதும். நான் மாத்தி ஏதாவது ஒண்ணு ஒரு தெரிஞ்ச போதும். நான் ஆஸ்தி பண்ணல, அது எப்படியோ போயிடுச்சு.
Hello, hello.
ஹலோ மேம்.
So have you noted this active site?
எஸ்.
Okay, so we
எஸ்.
will see the next question. So which of the following is correct for hemoglobin? So the option A is the binding with oxygen is weaker in comparison with myoglobin. That is true, right? So,
ஹலோ.
hello?
ஹலோ மேம்.
Am I audible?
எஸ் மேம், ஆடிபிள் ஆடிபிள்.
Okay. So the which of the following?
ஆடிபிள் மேம்.
Okay. So which of the following statement is correct for hemoglobin? So the option A is binding
ஃபர்ஸ்ட்
with
ஒன் இஸ்
oxygen is weaker in comparison with myoglobin. So this is true.
ஓகே.
Iron is also 5 coordinated. This is also true. So the
ஓகே.
oxidation state of iron is
ஓகே.
plus 2. That is also true. So all of the above is right. So option D is the right option. All of the above
ஆல்
is
த அபோவ் ரைட்.
right. You can note down these points.
ஓகே. ஓகே மேம். காப்பி. மேம், ஒன் டவுட் மேம்.
Tell that.
That diamide is that sorry, what is the SOS SOD, and the superoxide dispute is what is the role, ma'am? That is two role you told. That is superoxide radicals is catalyzed.
So superoxide dismutase and other thing.
What is the role of function of superoxide dismutase?
Because superoxide is not good for our human body. Now
The
water,
function means catalyzing the dismutation of superoxide radicals or reducing hydrogen peroxide.
it is reducing the superoxide anion into hydrogen peroxide.
Peroxide, okay, and not reducing hydrogen peroxide.
So
It's not.
reducing hydrogen peroxide into water is the role of catalase.
Oh, catalyst. Okay. 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 Thank you, ma'am.
Hmm.
Okay. Next question we can move, ma'am.
Okay, fine. So the metal involved in alcohol dehydrogenase are.
Zinc. Zinc.
Yeah. Yeah.
Liver alcohol dehydrogenase.
Yes, yes. Both in liver alcohol dehydrogenase, carbonic anhydrase, and carboxypeptidase.
This
Three
also.
enzymes are there in which zinc is
Yes.
present.
Yes. Yes.
And also the
Yes.
oxidation state of zinc will be plus 2
Two
always.
plus. Yes.
Okay, so now I showed you the reaction,
This
right?
is okay. Second one.
Yes,
I think
yes.
so. Second one. Yes.
Second one.
So this question. What is
Huh?
that? Assignment question.
Okay.
Assignment question.
Okay. So this is the this you have to remember.
Oh, yes, ma'am.
So which of the following statement is not true? This I will not tell. This you have to tell.
I think Yeah. That contains iron and magnesium. That's it.
Ah, yes,
Only
because
iron.
only
Iron is there.
iron is present,
Hmm.
right? You see, we are calling it as monooxygenases. Cysteine is also present,
Yes.
and many cytochromes are present.
Yes.
So this also you have to note down.
Yes.
So which biologically active species control important cellular process such as oxidation reduction, hydrolysis, and methyl
Metalloproteins. Metalloproteins.
metalloproteins? Very good.
It's a second question.
Okay.
That's why I'm answering.
The metal ion present in the active
Zinc.
site.
Zinc.
Oh, very good. You have remembered all the assignment question.
Yes, ma'am. Yesterday only I prepared.
Yesterday only you prepared.
Yes, ma'am.
Okay.
Once again, I'm reviewing all the assignment questions.
Hmm. This you can see. This you can note down at least actually.
Yes, ma'am.
You note down. I will wait for some 2 3 minutes. You note down.
That just I'm printing screen and then having in my desktop.
Okay.
Next, ma'am. Can move to next one.
So siderophores are small polydentate
Iron,
living
iron, iron,
with
only
high
seven.
iron affinity.
Iron, iron,
Their role is
oh,
to aid
iron transport or iron storage.
iron uptake.
I take.
Iron
So,
uptake in the organisms.
take.
So what is the magnetic nature of deoxygenate hemoglobin?
Deoxygenated hemoglobin. Diamagnetic.
Yes, diamagnetic. So hemoglobin in which oxidation state it is present? Fe
Uh, Deoxygenated state
plus 2.
22 plus. So 3D 9 10. 3D 10 I think so.
D 6.
10 for D6
So
also.
1, 2, 3,
Mm.
4, 5, all are paired. So it is
All are
diamagnetic.
bad.
So this
Rubridaxin,
read
riskin.
question properly.
Which of the following are examples? Not illa.
Which of the
Iron
following are
sulfur protein.
example?
Iron
Reproduction.
sulfur proteins, rubridaxin.
Yes.
But risky proteins, aconites.
Yes.
All the three except methane monoxidase.
Monoxygenous. You have to
This
read the question. Sometimes they will ask note
yeah,
iron sulfur.
assignment questions it's given. Not iron sulfur proteins, methane monoxidase.
Yeah. These things you have to read properly. Okay.
Yes.
Okay. So when in Fe2S2 cysteines are replaced by histidines, we will call
Oh,
it as risque proteins.
oh.
You note down this will be important for competitive exams also.
Okay, ma'am. Thank
So noted.
Note it down, ma'am. Note it
Which of the following is a copper transport protein?
down. And that is ceruloplasmin.
Plasmin. Yes. Most abundant element by atomic percentage.
Oxygen.
Carbon.
66.
Abundant
Human
element
body.
in human body.
Oh, oxygen is given 66 percentage.
Carbon is abundant.
No,
You
ma'am, it's
check it
given.
once again. Carbon should be the answer.
Oh,
So which of the following are example of copper containing enzyme?
and it's galactose oxidase.
Yes. So carbonic anhydrase has zinc, urease has nickel, catechol dioxygenase has iron,
And
galactose oxidase has copper. So you
uh,
have to note down any of
copper.
the like these all four enzyme and all
Okay,
the active centers
okay.
we have mentioned. You just note down it. This also you note down the active site.
Okay, ma'am.
So the oxidation state of iron in methemoglobin is
T, I think. Yes.
+3. So this key point you note down. Methemoglobin also known as ferri because iron in +3 we will call it as ferric. So ferrihemoglobin, it is a type of hemoglobin that can't bind or carry oxygen to the tissues. It is a metalloprotein
Oh,
in which the iron in heme group is in the +3 state instead of +2. Because in normal hemoglobin +2 is present, here +3 is present.
oh.
So well known, this is important.
B12, it's known. It's known.
This also I
132, it's
explained you
also
many
there.
times.
Already we have discussed.
Yeah, this you take the note of
Yeah,
this.
chlorine, chlorine, everything.
So storage of iron.
Ferritin. Yes, ma'am. Ferritin is also noted already.
So hemoglobin, function of hemoglobin.
Transport of oxygen.
Which of the following electron transfer proteins contain inorganic sulfides?
Inorganic sulfate. Inorganic sulfide வந்து peridoxins.
Rubridoxin,
Rubridoxins also.
ferredoxins.
ரெண்டு rubridoxin and peridoxins.
Not rubridoxins.
Oh, only peridoxin.
Because rubridoxin only one iron is present and one iron is attached to cysteines, all the four cysteine and cysteine sulfur are the part of organic sulfur, not inorganic.
So
Okay.
rubridoxin Fe0S.
Fe1S0.
One Fe is 0, S is 0.
Yeah, yeah.
No S.
No S means I this I told you many times, right?
Yeah, that's a confusion.
This is rubridoxin Fe1S0 because here S is 0 because and not any inorganic sulfide. All
Oh.
are from cysteine. So whatever
But
sulfurs
if,
are come.
inorganic sulfur is also absent, we can call it as iron sulfur protein. Shall we call it as iron sulfur protein?
Yeah, yeah. Sulfurs are there, but not inorganic sulfur.
Inorganic sulfur. Okay,
Their
okay.
names are based on inorganic sulfur. You
Okay,
see, if
okay.
here iron is
Inorganic
coordinated to
sulfur.
four sulfurs, but we will
Okay,
not
okay,
call
okay,
it as
okay.
Fe1S4
Okay, okay.
because no. Here, if you will
Only
see,
one Fe.
what
Okay,
is the
okay.
name
I
for
understand.
this? You tell me first.
Okay, okay, okay. Understand, understand.
No, no. First, you tell me the name for this. Then we will move.
Or what the name? Name means Fe4S4 like that.
Yeah. Four.
Fe4S4 is peridoxin.
No,
Another peridoxin is Fe3S2.
this is Fe2S2. What is the
Okay.
name for this? In the term of
Fe2.
number of iron and sulfur.
Fe2S2 means 3.
No,
I
no.
can't.
This you tell.
Aconitis, that is
Okay.
Fe3S2 aconitis.
Fe3S4, this is not S2.
Fe3S4 is
Aconitis.
hmm,
Okay.
sulfur.
How is acid base catalysis a complete
Through metal ions built into protein structures.
yeah. So
Second,
nature of myoglobin monomeric
my
tetra.
monomeric.
Okay.
Hemoglobin tetrameric.
What will be the
The
electronic
first one. First one.
first one?
Uh, Ma'am, deoxyhemoglobin, what is the structure, ma'am?
2T2G6 EG0.
Oh, that's one. Okay. The last
What is the nature of oxygen binding curve for myoglobin?
This is I know that binding curve of myoglobin is hyperbolic.
Yeah, for hemoglobin.
Hemoglobin is sigmoidal.
Yes.
pH dependent. This is pH dependent.
You note down this.
Uh, I'm having this curve. Everything's.
Okay.
Now one question to answer. I need that one. 1 minute.
Which metal is present in carboxypeptidase A?
I'll see zinc.
Yeah.
Ma'am, 1 minute, ma'am.
Huh.
That in which biological process is the iron dioxygen complex in hemoglobin involved?
In which biological process? Iron deoxy.
Process iron dioxygen complex in hemoglobin involved respiration. Is a transport of oxygen is there. Photosynthesis or respiration.
Iron dioxygenase, na?
Iron dioxygen complex in the hemoglobin.
In
Iron dioxygen complex.
transporting oxygen in the blood.
Yes, ma'am. The respiration. Is it respiration?
So, huh, it will be respiration only, I think.
Yes. That's it.
So
Okay, thank
this you have to note down the active site of carboxypeptidase A.
you. Okay, zinc. I noted.
Which of the following statement is not true about cytochrome P450?
It is a colored cell.
Yeah.
It's
So
that
this have important role in metabolism. Iron also it has
and then
involved in
sulfur.
oxidation. So
Yes.
it is a colored cell.
Epoxidation.
So this one,
This is the doubt I'm having.
doubt.
Active sec.
This is
Yeah, I'm
this is
remembering. 1 second.
this is methane monooxygenase.
The someone now only given FeOFe. First one, I
Yeah.
think so. Just I want to remember all these things.
Yeah, so I have discussed whatever I have prepared. If you have any doubt, you can ask.
I'm once again.
Many questions we have discussed today.
Yeah, ma'am. I'm very happy today.
Hmm.
Ma'am, iron sulfur clusters in biological system,
hmm.
they play a role in electron transfer.
Hmm. They
Uh,
are electron transfer proteins. We will call it as electron transfer protein.
Iron sulfur proteins are electron transfer protein.
Electron transfer or electron transport, anything.
Okay. Okay. That's all. Okay, ma'am. I'm also having class now. I'm moving.
Okay, fine. Okay, bye
Thank you,
bye.
ma'am. Thank
Okay.
you for your help. Really, today only I'm very happy, ma'am. So many questions we have discussed. I'm having some confidence.
Yeah. Yeah, in
Thank you,
between
time.
also, if you are feeling any doubt, you can contact me through my mail ID.
Okay, ma'am. I'll contact you.
Yeah. Okay,
Okay, ma'am.
fine.
Thank
Bye
you.
bye.
Thank you very much.